So, in this video, we're going to be talking about gene transfer. So, gene transfer is when genes are passed from one organism to another organism. The type of gene transfer you're probably familiar with is what's called vertical gene transfer. That's when my DNA is passed from a parent to an offspring. And this is the kind where, you got genes from your mother and your father, and they came together and made you. Bacteria accomplish vertical gene transfer through binary fission. So they replicate their DNA and then they pass it down to their offspring. In horizontal gene transfer, DNA is going to pass from a donor cell to a recipient cell. In this case, we produce what's called recombinant DNA. That's DNA from multiple sources. So, vertical gene transfer, parent to offspring. Horizontal gene transfer, donor to recipient. It makes recombinant DNA. There are three main types of gene transfer that we're going to talk about: transformation, conjugation, and transduction. We've actually already talked about transformation before, so that's when we're going to begin. So, horizontal gene transfer, transformation, this was first observed in an experiment performed by a scientist named Griffith. And he was studying two strains of pneumococcus. He was looking at a rough strain of pneumococcus and a smooth strain of pneumococcus. The rough strain did not have a capsule, and when it was injected into a mouse, the mouse survived, and when he cultured the blood of the mouse, he recultured out the rough cells. Remember Koch's postulates. We need to retest at the end. But when the smooth cells, which have that capsule, remember that's an important virulence factor, was injected into the mouse died. If you recultured the blood, you got smooth cells. If you took the smooth cells and heat killed them by boiling them and injected them into the mouse lived, and we didn't culture out any kind of bacteria because they had all been killed by the heat. But this is where it gets weird. If he took the heat-killed smooth cells and living rough cells, or R cells, and combined them together, then injected them into the mouse died, and he was able to reculture both smooth and rough cells. So this is really weird. What he concluded happened was that the heat-killed smooth cells were able to transform the rough cells into smooth cells. He didn't know what the transformative principle, what he called it, was. We now know that that transformative principle is DNA. So what was happening was when the smooth cells were heat killed, their DNA got released into the environment, and then the rough cells were able to pick up that DNA, and that was able to transform them into smooth cells. So we call this ability to take up DNA from the environment transformation. So in transformation, DNA in the environment, which is usually going to occur when a bacterium dies and releases its DNA, is then taken up by a living cell. So, how does transformation work? Again, transformation, this is when we take up DNA from the environment, and the donor is usually a dead cell, like those heat-killed S cells. We call cells that are able to be transformed in this way competent cells. Remember, we made our cells competent when we did the pea glow experiment. So competent cells are cells with a high rate of transformation. The cells that we used in pea glow were not naturally competent, so we had to use the heat shock and the calcium chloride. Cells that are naturally competent have what are called competence factors. And these competence factors are these membrane-bound proteins that are able to attach to DNA in the environment and actually pull the DNA into the cell. Once that DNA is into the cell, The cell actually looks for regions of what are called homology, areas where the two pieces of DNA are similar, and then they use that to actually insert this new environmental DNA into their chromosomes. This can also be done by the cell taking up a plasmid like we did in lab. Okay, so that's transformation. We're going to take up DNA from the environment. The source of the DNA is usually a dead cell. If a cell is naturally competent, it's going to have competence factors, which are these membrane proteins that bind to extracellular DNA and take it up inside of the cell.